there too.
Oh, yeah. Let me get a good focus.
One of the things I'm excited about as the education and outreach coordinator for the sanctuary is to
And that black coral there right at the lasers?
bring this imagery back and these communities back to people and work to figure out how to connect people.
Oh, there's a
that?
little jelly at the... Neat.
Ooh. Wow.
That's cool.
That's some nice buoyancy. I'm excited to figure out ways to bring this back to people so they can learn about these habitats and that they are so vital to biodiversity and connected to us. We've seen some trash in the canyon and some of the other deep sea explorations, and we have an impact down here.
Yes, and I think that education can play a really big role in having a positive impact in helping people connect their behaviors.
How about that red shrimp to the right of the lasers?
And helping people connect their behaviors to our waterways.
Give him his...
You guys let me know when we're ready to move on
4 minutes
with the ship and ROVs.
Yeah, the shipper's moving just at this depth. Everything's a lot slower.
Okay.
Okay, coming wide.
Which means if you guys want to sample, it could take a while to get set up and back to where we want to be.
Okay, thanks for that logistics info. It's helpful.
Yeah, I'm starting to move them away. But we may need to come up just a little bit.
Can we look at that white stalked organism below the lasers, straight down below the lasers, white at the bottom of the screen now?
Next to the peach?
To the right of that peachy one.
Yeah,
there you go. It's kind of center. Yeah, right where the lasers are. Is that another?
I'll let Dan...
I think it might just be another black.
Okay, zooming in slowly.
It's really fun on this expedition. We've been able to really compare and contrast habitats. We were in shallower waters for a couple days up in south of Point Arena, and now we're back in the deep.
Is that the one you wanted?
Yes.
Okay.
And this is our final dive in Cordell Bank National Marine Sanctuary and Greater Fairlands National Marine Sanctuary. So we're really excited and hoping that we see some amazing animals.
Yeah.
On
it's all right.
You good? On nautiluslive.org, though, you can see our last expedition of the year. That will be in Monterey Bay National Marine Sanctuary. And that's just coming up next week. We have one question from someone tuning in about how the canyon was formed, and I wasn't certain if Guy was listening and able to answer that.
Okay. Okay.
Hey, Guy, do you want to take a stab at that question about how this canyon was formed?
Whoa. Nominally it's supposed to be 0.5, but 0.2 to 0.8.
Well, not really, but essentially there's been uplift of the California coast for millions of years, actually. And when you uplift something, eventually gravitational forces are going to cause landsliding. And so initially you have
Oh, what's that?
landslides spread out along the slope, and they gradually sort of coalesce into
Yeah,
a evenly spaced system of canyons.
Could we back off a little bit?
Mm-hmm.
That's helpful.
Mm-hmm.
And Guy, weren't we talking that maybe this canyon may have been connected to San Francisco Bay at long time ago with sea level much lower, just because we see that little curve that comes up on the continental shelf?
It's possible. I don't know if anybody's actually established that, but it does look like the faulting has basically shifted it north relative to the bay. And there must have been some sort of
Can I get a zoom on
drainage
that
pattern
C-pen that's
coming out of
coming
the
up
bay
just to the
across the
left
shelf.
of the lasers and now just below the lasers? Is that maybe another,
The top-down view isn't always the
Yeah.
best for a C-pen.
you're right. Let's keep,
But
we can zoom out and keep going.
yeah,
Looking for
they're coming
another
wide.
one.
Yeah.
I see a little rocky outcropping at the top of the left screen.
Oh, yeah. Mm-hmm.
Danny, would you be able to share with us some of the research objectives for this particular dive?
Sure, this dive is the deepest we've ever been to in Cordell Bank and the deepest portion of this canyon. So we're really exploring, seeing what's here, because we expect that there might be some, and we have seen some already, some different species than some of the shallower dives that we've seen. So part of our goal is to inventory species in the sanctuary so we can understand the full complement of species and communities in the sanctuary. And we're also, because we have now surveyed some of the other areas in this deep habitat in the sanctuary, starting to put together some of the idea of the zonation in the sanctuary and how communities change through these depth zones, which tends to structure the deep sea communities. So we were planning to be in exploratory mode and zooming and possibly collecting, and we will be trying to move our way up the canyon for as long as we have. We have about, let's see, we have about probably another 6 hours or so to explore. So we'll be trying to cover as much ground as we can. And can you zoom on the bottom left? There's something on a stalk there. Is that just another Umbellapathes? Yeah. And that one seems like it has 4 fronds. Okay, you can zoom out.
Okay. Just another black coral.
Yeah.
Ho-hum.
Looks like Umbellapathes.
Is it true that squat lobsters are actually crabs?
I think I have read that before. I'm not an expert though. I think I can look that up for you.
No problem.
What's that stalked just above the lasers? There's just at the top of the screen, the top view,
The
maybe
little
a
tinge of
worm
coral or
tube, I
pink.
think.
Oh, yeah.
Yeah. Or maybe a tube anemone.
Thanks for tuning in, Kathy, Terry, and Carol in Wilmington, North Carolina. Appreciate you guys tuning in.
So, according
can
to
zoom out.
NOAA Ocean Explorer, squat lobsters are actually more closely related to hermit crabs.
Oh.
They are all in the arthropod family, though. We think of them as scavengers, but they have been observed catching fish from the water column. That's pretty cool.
I used to feed one frozen shrimp. It would reach up and grab it out of the forceps. It was great.
Aw.
I guess you got to do what you got to do if you got to get food.
Don't.
What is that? Can we get a zoom on that? I thought it was a predatory tunicate at first, but now I don't know.
It looks like it has
Bizarre.
like fangs or something inside.
I know.
It's a Halloween special.
I wonder if that could be a benthic ctenophore on something.
It does look like it's illuminated from the inside.
Yeah. Weird.
Something that ingested.
That's what I was thinking, Jan. Looks a little different than the ones we've collected before.
Do we want it?
No. Okay, I don't
Okay.
think we'll want it. Unless we
Okay.
Let's
Come
see if
again.
we see some more of those.
What are some species of interest that that you hope to collect on this dive, Danny?
Well, because we're in this deep depth range, we were thinking there could be some black corals that we're interested in. We've already have seen and collected some of the black corals. And also we're looking for some Chrysogorgia corals, which we haven't seen yet because they're really deep. And let's get some zooms on these sponges.
Okay.
These white. Those are cool.
These are different.
Different.
Yep. Let me take a look.
It looks. Are those some of those eggs?
Let's get some good pictures of this, and I think we're going to get a sample if you're in a good place.
Okay. Did you want a sample of this?
Yes.
Roger.
Can we get a picture with the lasers off by some chance?
Roger.
This looks like it could be the heterocone, but we haven't seen one like this before.
Unless you want me to step back.
All right. That's full
And those
zoom.
lasers that
Roger.
we just turned off, that is a way to consistently have a 10 centimeter measure to help us understand scale.
Okay, got some good pictures, thanks.
Okay, coming wide.
Oh, you could do lasers back on.
Can really see the marine snow moving by, which is stuff that plankton and dead stuff from the surface waters that rains down and is food for these sponges and corals.
Mark where this is in case you get pulled off.
Sure.
Dan, do you want the lasers on?
Science, you want the lasers
I would
back
like the lasers on, please.
Roger.
Jenny, connected to the marine snow, one of our viewers was curious, when an animal in the sea dies, does it float, sink? How does it kind of break down to be part of that marine snow?
Well, I think it depends on the organism. Some things probably float for a while and are decomposed by predators up top. We've seen white sharks scavenging on dead whale carcasses and seabirds scavenging on whale carcasses. So there's a bit of scavenging at the surface. But eventually, everything's going to sink to the bottom, whether it just be the bones or the blubber or whatever's left. When it becomes negatively buoyant, it's going to sink to the bottom at some point and may decay midwater, and then eventually when it hits to the seafloor, there's a whole ecosystem around whale fall communities and different worms and hagfish and ratfish eat on those whale fall communities. They're pretty interesting. But all those nutrients are reused by animals on the seafloor, and in some cases near upwelling zones, upwelled to surface waters to act as fertilizer for phytoplankton, which is the base of the food web in the springtime. When we get that influx of nutrients and we have longer days of sunlight, we get phytoplankton blooms, which are the basis of our really diverse food web here on the coast. The surface waters and what's happening up top are directly related to the health of the bottom.
50 to 60 meters of swing in Argus that I got to overcome.
Since a lot of these all the corals and sponges rely on all that stuff from up above. So if we don't have all that plankton and all that life up above, everything down below doesn't get food either. So a lot of interconnection there. And one
depth,
species that we sampled already during this cruise, but are always looking for our bamboo corals, and they actually can record in their layers how much food they've eaten. They give it like somewhat of a signature of the condition of the environment when they grow. So bamboo corals are an excellent time capsule to study the health of the ocean. And because they are long-lived, it can live to about 400 years, that we know at least 400 years, we can study them and get a time capsule of what our environmental conditions were 400 years ago. A student at Bodega Marine Lab, Karina Fish, who's also a Nancy Foster scholar, a NOAA scholarship, is studying those corals and connecting both calcium carbonate chemistry and how they're doing at depth with the chemistry of the water, but also looking at that historical timeline based on the records they're keeping within their skeleton. It's a really fascinating species.
Definitely. Thanks for sharing all that. And to help people understand, a bit of a pause right now. Our ROVs, they are tethered together. So Hercules is the ROV that's capturing all this amazing footage. Hercules has 7 cameras, in fact. But Hercules is tethered to another ROV called Argus. Argus sits a little bit higher in the water column and helps to typically take on a fair amount of that energy in the water so Hercules can maneuver well and collect fragile samples often. And then Argus is tethered to our ship, the EV Nautilus. So for us to take a sample, we need to be well positioned, we need to be stabilized, and all three, Hercules, Argus, and the EV Nautilus, need to be in position to do such. So because we're at 3,000 meters, our navigator Miles told us at the beginning of our watch that it would take a bit of time to maneuver the two ROVs in the ship into place so we could take those samples when needed. And Miles, feel free to add on.
Yeah, that's all correct. Just sort of dealing with where I got myself into.
So you see the Argus view is lost for a moment. So Antonella has kindly spun 180 just to give us a little bit more tether while we move the ship. Because I was, Hercules was getting tugged off there.
Yes, so
might give us enough to stay on it until we get the ship in position.
Okay.
Can't see Hercules anymore, but we know where it is. Since we're not going anywhere, we can get away with that because not have to worry running into a cliff since we're not moving.
So the quad view does show you Argus's video, correct, Wayne?
That's correct. Sat feed 2 is the view of Argus, or our
From
clump
Argus.
weight vehicle, which is, as you mentioned, higher in the column and absorbing the kinetic energy of the waves of EV Nautilus and isolating those from Hercules, which is on a much more flexible tether between Argus and Hercules. Normally we like to keep...
I think the two on the right might be the same.
Normally we like to keep Argus positioned in a way where we can keep an eye on Hercules, so we can see how close he is to rocks like the one in front of it right now, and
what's that?
Any potential dangerous edges. But what Dan was referring to was the fact that they've rotated Argus so that the end that has the tether attached is actually using the length of Argus, the vehicle, as an extra length of tether so that he can get a bit closer while we reposition the ship. And once the ship's in position, we'll be able to spin Argus back around, and our satellite feed 2 will
good tether now.
Have
hopefully
Hercules in frame.
Nice.
Like we normally do.
And what are the two other pictures in the quad view?
Sat feed 1 is the main Hercules Argus camera, and sat feed 3 is currently bubble cam, and that is a downward facing camera. It's pointing straight down on the front porch of Hercules, so you can see a bit of the porch at the bottom, and that's the porch that would protrude out if we want to collect something. And the bubble cam is actually a PTZ camera, so a point pan, tilt, and zoom. So the ROV pilots have the ability to spin it around so it can look at different gauges and just to see, you know, the different parts of the ROV. Right now, this is where the stored position is, or the stowed position. I got you there.
You're right at 40 meters. You're like 38 meters out on sonar.
This is just for in the van.
Just,
Yeah.
Yeah.
Nobody sees that.
Yeah.
On the fourth panel on nautiluslive.org is a map overview of the current location of EV Nautilus.
Got it.
So,
It's coming back to you.
yeah.
Thanks for walking us through that. And hi, Corey from Cambridge, Massachusetts. Thanks for tuning in.
Science, have you guys decided what sample you want here?
Yeah, I'd like to, this sample on the corner that we're right in front of, and I think we have 2 species here potentially, so I'd like to get 2 samples. Also, since it takes us a while to get settled, that'll be more efficient. So,
Yeah, we're still moving the ship around, but I think I can, if I know what you want, I can think about where we're gonna
So
park
that-
Hercules to get it.
that top one, and then the smaller one that's just below it towards the left.
Okie doke. Can you
Come
look
wide.
at the, no, Anatola can look at the left side of the porch with the bubble cam? I don't know if it's worth parking or trying it mid-water it. I have good luck with those mid-water ones on a sponge.
Looks like you have a decent cat in butt cam. You might be able to turn around now.
What's that?
Argus may be able to spin.
Yeah, Rich. I'd rather have the leash now, because if we take a heave, we'll... Oh, that camera is slow.
And hi, Vivian from Alameda. We're doing a 12-hour dive, and this is actually our shortest dive of our expedition out in Cordell Bank National Marine Sanctuary in the Greater Farallones National Marine Sanctuary.
Very soft.
We've
should've
done dives
perched
that are
on the
typically
rock.
around 24 and 22 hours.
Do you notice the stalk on that sponge? Yeah.
Cool.
What are you noticing, Taylor Ann?
Taylor Ann was pointing us towards this.
Wayne,
These
can you
sponges
zoom
are
out for me?
stalked.
Thanks. Just using that bottom left corner to watch the perch there.
Okay.
So some sponges are stalked, and some kind of, you know, they grow right onto the rock. So this is stalked, which was hard to see when we were out in front of it. So it's nice to have this side view.
Yeah.
We get some good photos there
Okay, you can zoom
of
right in there, Wayne.
both the top and
Zooming in.
bottom species.
Nice. Yeah,
Okay.
the large one in particular, I would have missed that if you didn't point it out, Taylor Ann. Thank you.
No problem.
Sorry about the dust, guys. It's a little soft there on the landing.
It's all right, we're here.
And yeah, when we sample, we typically land just to be as stable as possible because Herc has these amazing manipulator arms, but it's a pretty fine science to make these cuts of these biological specimens. So that's why you're seeing a bit of sediment floating up.
Bad pilot. grip force. 3.
Can we get one quick picture without the lasers first?
Sure.
And then turn them back on after?
Me-
Do you want to? Can you kill the lasers for them?
All right, thank you.
Okay.
Oh, that's nice with the dark silhouette,
Yeah.
It's beautiful.
dark
BBC
background.
shot. Except for that
Yeah,
dust.
it's
Give it a minute. It'll be gone.
a difficult shot to get the right amount of light because it's so white and half of our screen is so black.
Since it is such a nice shot, if we could just hang for another 30 seconds or so and see if we get some of that dust to go away.
Okay.
So again, this is our deepest dive. We're at 3,000 meters.
Could you tilt up just a tad so that the top portion of that sponge is in frame?
I can come wide a bit.
There we go.
yeah.
Well, you know, there's just more mud coming, so it's not really,
Yeah.
we're not going to get a clear shot. So we can just go ahead and sample, and I'd like a grab of each one of that top and the bottom one. It can go into a slurp or a small box, but they should be in 2 different containers.
Oh, looks like we got.
Yeah, we have 2 large boxes left and then 3 of the regular size, smaller starboard boxes. But if you did think they needed 2 large boxes, they could each be separated.
Do you think they will those float or sink?
Hard to say.
I could snip off a piece and feed it to the slurp and it would go in the jar, or we could try and slurp, but I don't know if how friable it is, if it'll break away.
I'm not sure either. Last sponge we sampled where we grabbed a piece with the claw and broke it off, that seemed to get us a nice sized chunk.
Yeah, I could do that.
Okay. Taylor Ann, how many slurps do we have left?
We have only collected 3, so we still have 4, 5, 6, and 7.
So you want a decent sized piece of it, do you?
Yes.
Something like that?
Yes.
Copy that. Hope his stock doesn't break. Might get more than you bargained for it.
So Herc's hand has a scissors. Herc's hand can rotate 360 degrees. You're seeing a lot of those abilities in this one sample collection.
Nice. And it looks like it sinks.
Okay. That might be a bit much for the little box. Let's go for Delta.
We collect the sponge so we can get a positive ID on it by sending it to sponge experts. They have to look at the intricate pattern of the spicules.
Can you zoom out, Wayne?
Coming wide.
Thank you. Okay.
Full wide.
You want to open Delta? Open the front?
The filter?
Yes, please.
Roger.
So right now you're seeing
And
the
there's
porch
a porch
of
light.
Hercules.
There's a porch light, yeah. Should have it on your GUI now.
There
thank
we
you.
go.
Beautiful. This is one of our sample boxes.
So it can't go in the left one, right? Needs to be separate from that pen? Or can
well,
it go in with the pen?
Since we have room in Omega, I guess we could put it in there.
On top of the whip?
Oh, I was saying the one that you're above right now.
Roger. Okay, ready to close?
Ready.
Oh, come on, be gentle on the box. Okay.
Mm-hmm.
Nice job.
Nice.
Taylor, and you have that as sample box Omega, or?
Yep, sample
Okay.
box Omega, and that's sample number 50.
Thank you.
Taylor Ann, can you explain what goes on in the wet lab once we bring our ROVs up and collect our samples?
Sure. So once we get all of the samples up to the wet lab, we take them off of the ROV first, and then we put them into the refrigerators in wet lab to keep them at a constant cool temperature while we wait to process each of the samples, giving them their proper ID number, labeling them, taking off any associates that might be on there, because that can tell us a lot about the organism we're collecting as well, like what other animals that might live on it. And then once we process all of those samples, we'll send them off to certain labs that specialize in identifying them and do some further research on them. So we can collect anything
Go
from
for full zoom there if
sponges
you want, Wayne, while we're
to
waiting.
corals, sediment cores, water samples. But we're particularly interested in the sponges and corals on this dive here. We're seeing some really interesting things today.
Do you want the stem on this guy too?
No, I don't think we need the stem.
So just,
Yeah, just the same kind of thing you did
Okay.
there. I know you can't help how it breaks off, but that was we don't need quite as much material as we got. So if
yeah, this one's small enough I can probably use the coral cutters and hold it in the...
there you go, yeah.
Wow, it's bigger than it looks. Something like that there.
Sure, let's give that a try.
It can be less or more if you want.
Let's try a little bit less. There, let's try that.
That's not going to be a lot
No,
there.
not enough. Okay, let's.
It's just going to be
Yeah,
like
that's
that
a
little
tiny little
bit
snip,
on
yeah.
the blade there.
Let's try a little bit more then.
There are a lot of different sponges in this section. I'm just looking through and looking behind and under, and there's like I think it looks like a glass sponge up there. It's kind of tube-like with the
Yeah.
spicules sticking out.
But
And-
still going to be a very small sample.
Okay, you want to try getting a little bit material in the claw there?
Yeah. Yeah, it's bigger than I thought. I don't think we'll be able to use just the blade. That's like too much now. I can't make up my mind.
That looks good. Let's go for that.
Okay.
Danny, what are these lollipop-like things sticking off? It's like
I
a
think
stick
those
with
are a sponge as well.
Those are interesting. I'm not sure if I'm seeing a tube worm behind them, but it looks like they have something that's like casts out like silicates or some
that
type
help?
of a web that catches stuff, or I'm not sure if it's something behind them.
This guy doesn't seem as friable as the other guy, so
Okay. It's not going to cut?
it doesn't look like it. I'm
Okay.
probably going to break the stem there, and you're
We
going to wind up with the whole animal.
Well, let's try turning it and see if it breaks off, and if it all comes off, then that's what we got.
It's coming.
Okay.
Mm-hmm.
Oh boy.
That stem was more,
A little more sturdy.
yeah, it's,
There it is.
Taylor,
Okay.
Ann, when you see this, do you think about all the associates for processing later?
Now, is this one going to have to go in with the lambda, with the whip, probably? It's our
it would
biggest
be
one.
nice if it did. Yeah, it would be easy for us. The problem with the starboard box is we have to disable the verts, and we're on perched on a marble here. So, I mean, I could float up. It's probably all right. But I think he's a little big to go in one of the small boxes. So yeah, if we can put him in the front with the whip, that would be awesome.
Okay, so it won't fit on the side drawer?
It will, but not, I don't think it'll fit in one of the smaller ones.
We do have one big one left on the side drawer.
Okay, let's do that one.
On the side one?
Yes.
Okay, coming wide.
Roger.
We're going for a flight.
Woo! And that'll be starboard box F.
Starboard box F.
And that's sample number 51.
Let me know when
All
you're
right,
ready for
I'm
me
going
to
to
secure
change
the
our
thruster.
set feed.
Do you want laterals and verticals?
Yeah, the problem is we'll start floating then, but the current might keep us pinned in, so
Okay. Tell me when.
I'll get around there and we'll get ready, and then we'll turn it off before we open the box, because I don't know what's Pandora's box with all those samples in it. I don't want the stuff to come out. Didn't Greg mention you could kill the starboard light?
Yes. Do you get that?
Woo! Yeah, you can. You can kill that vert now.
Okay. Vertical. Vertical thruster secure.
And we're floating.
Should
Go ahead and turn the thrusters back on, please.
Thruster on.
Yeah, let's get situated here. I'll just okay, turn off the starboard vert. See if I can pin it with one vert.
Okay, it's off.
Copy.
Got an enemy there with the really long tentacles. That's really cool.
Okay, where were we?
And all of our samples are shared with relevant experts and partners on these expeditions. And then
Think I'm high enough there?
biological.
Okay. Open, open. Yeah, don't open the little boxes. You said F, right?
Yep, that's correct.
That guy was bigger than he looked.
Yeah, that's a nice sample. Looks like we got a snailfish checking us out.
Hope he doesn't float. Okay.
Awesome.
Sweet.
Great. And just to finish that thought, all of our biological samples are all go to Harvard University's Museum of Comparative Zoology.
Roger.
Are we still kind of stationary here? We could.
I'm kind of swinging in
Okay.
there. I disabled the thrusters, but we're...
All right, that's all right. I was just going to say if we were still sitting, we could zoom around and look at this little corner because there was lots of small stuff there. But
yeah, I can perch back in there.
We should be stable enough to try and turn Argus around.
Yeah, you can turn Argus if you want.
Okay.
First, Our sponge and
that
coral
Bert back
samples
on,
will
is it?
actually be
It
going
is,
to the
yes.
California
Thank
Academy
you.
of Sciences. Gary Williams is with us, who's a coral expert, and will be identifying the corals and working with other sponge experts to get our samples to some sponge experts. But I'm sure some pieces will go to Harvard as well.
Yeah, we when the Cordell Bank and Greater Farallones National Marine Sanctuaries are doing dives here on the Nautilus, we do duplicate samples because all the samples go to Harvard for
Yep.
Nautilus, and then
Okay,
we do a
Wayne, zoom away.
duplicate sample for Cal Academy. So because that's a repository for all samples in the Central California National
Okay.
Marine Sanctuary. So we actually have a double effort. We don't collect any more material. We usually just divide up the material that we collect.
Sure.
So these little there's a lot of sponges on here, and these little, they kind of look like sticks with balls on top. Those, I think, are sponges, and they're kind of similar to one that we collected last cruise in 2017, which was, we thought at first was going to be a new species, but turns out it's a range extension from Russia.
And Danny, why do we sometimes take a portion of a coral or sponge, and other times we will take the whole animal?
Well, like in this example, we didn't need the whole animal, but it just came off, so that sometimes happens. We try and take the least amount of material that we can to be able to identify the species. Usually that you can do a portion of the animal, but sometimes you need to look at more than one feature on the animal or a branching pattern. That involves taking more material. Sometimes the samples are so small that by the time you get the manipulator around it, you're capturing the whole animal. So try and take as little as we can, but sometimes we get the whole one.
Do you see that worm tube in there?
Oh yeah, I do.
I wonder if
Behind
that's
there. Our similar to our worm tubes we saw the other day.
Yeah, I wonder.
Yeah,
What's
couldn't
that?
really see those fine hairs on the ends of those white.
Oh, now you can see them.
Yeah,
They look like lollipops.
they do.
Cake pops.
Can
Okay.
can you kind of zoom out a little bit and just let's look around here and see what else we can focus on.
Mhm.
Also, I think the lasers are not back on, are they? Okay.
And that anemone at the bottom with has those really long tentacles. Can we get a zoom on that?
Sure. Mhm.
I'm not sure what that is, but it looks a little different than other ones we've seen. I don't think we need to collect that, but I wanted to take a look. And you can zoom out.
Okay, coming wide. Full wide.
Okay, I was going to have us move along here, but I see we've got folks coming in for dinner swap. So if you wanted to do that before you lifted off this spot, we could do that.
Yeah, copy that. I'll ask Mike what he prefers.
And I'll zoom where we're at while we're here.
To those tuning in, we are organized into 3 different watches. So you're currently listening to the 4 to 8 watch, meaning we're on from 4 a.m. to 8 a.m. and then again 4 p.m. to 8 p.m. Pacific time. But in this 4 p.m. to 8 p.m. watch, we get relieved to eat dinner. So people from another watch come on and take over our role for 30 minutes. But there's 2 other watch teams as well, so we're able to work around the clock. It also means that there's 3 scientists, you know, three individuals for each role at least on the ship. So in total there's 31 scientists. and 17 crew on EV Nautilus that make this work all happen. But again, many of these expeditions exceed 20-something hours. I think our longest expedition this season has been over 60 hours. So to keep the ROVs flying and capturing all this amazing footage, it means we have to be on this watch schedule. That's a little behind the scenes info for you.
Hello, science.
Hello, Herc.
How's it going?
Good.
We're pretty great.
All right, we're not moving anywhere
Okay, so we can.
at the moment. I think the word was you were deciding to go ahead. Sample again.
We're gonna explore.
Okay. Are you on SPL?
Oh.
I can't really hear too well. Sorry.
Let's see.
She looks like she is.
Is that better?
Oh, the volume's just way down.
Okay. So we don't need to sample this,
Okay.
but we can poke around on this rock feature.
Sure.
They sampled these two sponges to the right.
Okay.
So
Do you want me to look up anywhere while we're sitting here? We got some nice still images we can push in on anything you want to see, or we can keep going.
let's look at that clear floaty thing that's attached to the orange
Fuzzy
anemones.
dice. Yeah.
Fuzzy dice.
Fuzzy dice.
Well,
call.
that anemone is beautiful. Wow.
And then he is beautiful. I can center it up on this guy a little bit, and you can push in some more of their video.
Mm-hmm.
Oh, hello, Wayne.
How you doing?
I'm doing well.
Wow.
Let's get that in focus. Stand by.
Tunicate? What do you think? It looks like it's hooked onto the anemone,
Yeah.
right? On that one. Oh no, it does
It
have a
has
stalk.
a stalk, yeah.
Oh, right there.
Oh, so this might be one of those predatory tunicates. But it does look different.
It might be the anemone's birthday, and it's a balloon.
Seems pretty promising.
Yeah, very probable. Thanks for that contribution, Wayne.
Hey, I'll be here all day.
Anything else you guys want to see? Or are you still thinking about that guy?
Are we waiting for a ship move or can we move? What's the
How is it attached?
We haven't called in a ship move
Okay.
yet.
but we're able to head to the left?
Yep.
Okay.
Wow, that is one.
You ready? Yeah.
Because they have spent some time on this rock.
Okay.
Yeah.
Okay.
Got moving then.
Yeah, we can cruise.
Bridge enough. Can I have a 20-meter step bearing 090?
Some of our viewers were super interested in learning a little bit more about Herc. Herc's pretty huge in terms of size. 3.9 meters long, that's 12.8 feet. About the size of a car, probably bigger. 7 feet tall, 6 feet wide. So when it's on the EV Nautilus, it looks quite large. And then when we deploy Herc to the ocean, Herc looks pretty small in the large expanse of the sea.
Mm-hmm. Mm.
Beautiful rock.
Yeah.
Are those more of those tourniquets?
Yeah, I believe those are tunicates as well. It's just so fun to see these sponges on stalks. I didn't picture that when we first saw them.
Mm-hmm.
Okay to zoom?
Sure, go ahead.
Okay.
If you want to hold there, I'm going to come
Yep.
in a little bit for you too.
Oh, that'll work.
There you go.
Not focus.
Get you some better light on it.
Mm-hmm. Thank you.
Sure. And I'm at the end of my tether here, so
Oh, okay.
we'll just hang out here for a moment. What are we facing? We're facing the other way.
So we think those are similar to the ones that we sampled?
Yes, I believe so.
Okay. Coming wide.
Sure thing.
All right, full wide.
All right, so where are we going? 090, correct?
Yeah, that's
Ready?
correct.
Okay, 4 to 6. I am going to relinquish my seat as video engineer to Ashley, so she can enjoy a few more hours on her last dive.
Welcome, Ashley.
Roger that. Thanks, Wayne.
Thank you.
Bye, Wayne.
All right, I'm starting to come over to 090. All this soup.
Bridge enough. Another step, please.
My goodness, look at us.
Oh, beautiful.
Wow, that's pretty.
Oh,
Yeah.
those are the ones you were looking for, right? Or no?
Well, these are black coral
Sure
called
thing.
Bathy Pathies.
I'm just gonna
And
continue
we were
this way.
seeing these in Box Canyon, but they had orange polyps. So we believe it's the same species, and that's actually
Okay.
one of the deepest black corals ever recorded.
Wow.
Was it 8,600 meters?
I think you're right.
Those were
Yeah,
recorded?
yeah. And that would have been collected by a dredge from a very long time ago.
All right.
And Caitlin, what species are you most excited to see in Bodega Canyon?
Well, so far, as soon as we landed, I was excited. There are so many, this is the first time we've ever been to this depth. And initially we landed on mud and we collected two soft corals, a sea pen,
Roger. Thank you.
and two black corals that we do not have collections
Yeah,
for.
sure thing.
So initially off the bat, I was excited to see everything.
Sure thing. Coming up slowly. I'm gonna face the slope a little bit more.
You're happy with 20-meter steps?
Yeah, that's fine.
Okay. Bridge enough? Another step, please.
90's fine. The slope looks like it's a bit off there, so as long as the gains are proper, I suppose.
I was also most excited for this dive because of the depth, but also being from New York City. Bodegas to us are small grocery stores, like convenience stores, your corner store. And so it just was funny to me that I'm going across the country yet visiting a bodega at the bottom of the ocean.
So on land, due east of where we are is Bodega Bay. And then there is a town of Bodega.
Got you.
Yeah. and
Which is
Thank you.
where the movie Birds was filmed, right? That bodega?
Bodega Bay, where Birds was
No,
filmed.
bodega.
Oh, it was, huh?
Yeah.
Looks like we're going slightly down slope a little bit. How about I'll come up here, actually, Mike, you're right, and we'll have Argus look at the slope. And I'll just ride a little high and lateral across the feature instead of going forward on it. better.